expecting to be at before 2, then don't feel embarrassed about eating before 2. I think we're coming along. It's amazing. I wonder how many places in the world you can turn out audience of 100 or so on Friday afternoon to hear about the brain. And some people would like to hear about whether science can explain consciousness. Well, of course, the answer is in principle it can, but you'd never know whether it had. I'm talking about the human brain. I'm just feeling very nervous being only a couple of metres away from Richard Dawkins, who undoubtedly terrified. But I can actually remember the first communication that I ever had from Richard in 1976, something like that. And I'd given a lecture. He wrote a very, very, because of course Richard is a penetrating, polite person, pointed out that I had some confusion between group selection and however else it was. Yes, which is still the case. Okay, so let's talk about the keys here. All right. Well, you all know the issue that human beings are special and distinctive in various ways. Many of the ways in which it's easy to say we're distinctive don't bear really close scrutiny because examples of similarity can be found in other species. But the totality of what human beings can do and the flexibility of what they do cognitively does seem to be distinctive. You know, we have this capacity to reflect on the past and think about the future, to plan, to be able to build models in our heads of what has happened to us and therefore what's likely to happen, models of other members of our species and other species and so on. Constructing a sort of internal computational model of what we're about and how we fit in the world. Sometimes. This is a photograph of a group of workmen who have just finished cementing these very stout So I give you a sort of 10-minute history of brain research and where it's been, where it might go. And being in Oxford, it has to start with this character who was in Oxford during the Civil War, that remarkable intellectual hothouse which led to the creation of the Royal Society. He was a physician to the royal family when the monarchy was reinstated. He was certainly one of the best-known physicians in Europe. And he was also a great observer. A great describer of disease and abnormal structure and function. And you can see that from the skulls that lit his desk that the brain was one of his particular interests. In 1664 he wrote a book called Cerebri Anatomy, which was used as a textbook until the beginning of the 19th century. Those of us who occasionally attempted to write textbooks, we can the textbook can be used for 100 years. And the drawings were magnificent. This drawing of the base of the human brain, undoubtedly a human brain, a real achievement if you've ever seen a living human brain. It's a sort of squishy jelly thing. It falls apart very easily. It's not fixed. So I've got it out of the skull intact. So it's really quite an achievement. The picture wasn't Lewis's. It was drawn almost certainly by Christopher Wren, who worked with Lewis and was the president of the Royal Society, was one of Britain's greatest scientists before he defected to the arts side and probably became better known in This is Wren's drawing of the base of the human brain. Now, Willis's writing is remarkable, and his inferences from observation were extraordinarily impressive to me, almost Darwinian in his speculations about the relationship between form and function and between different species. So one way in which he tackled looking at the function of the brain was to compare different species. And what basically the argument was there must be similarities in function between parts of the brain in different animals that look separate in different And that, of course, was already quite a sort of leap. And then secondly, to say that any bit of the brain which was unusually developed, species, small or large, must be correlated with any aspect of behaviour that was especially unusual in that species. So when you look at the human brain, you saw the obvious thing we all know, that the cerebral hemisphere, this massive folded stuff at the top, is much, much greater than in any other species. 4 times the volume of the chimpanzee cerebrum. Very close, genetically very close relatives. And that, by the way, I mean, just to anticipate the end of the talk, that's an interesting commentary on the relationship between phenotype, that is what we actually look like, and genotype, because the genetic difference between chimpanzees and humans, as you know, is very, very simple. And yet there can be enormous differences in appearance, particularly in the brain. Okay, so he argued, well, if it's so much bigger in humans, it must be responsible for things that are especially well developed in human behaviour and mental function. So he said things like perception and thought, reasoning, and so on. What was called estimation must take place in the cerebral hemispheres. Just the sorts of functions, memory as well, just the sorts of functions that we do think about cerebral hemispheres now. And by the way, in doing that, he also took another very innovative leap in thinking, because he detached himself from the theory which had dominated views of the brain since Galen from the 2nd century, that the tissue of the brain, the actual substance of the brain, is not important. It's only, the brain's only there, again, we would argue, and all of us, that the only function of the brain is to, the tissue of the brain, is to enclose chambers inside the brain, which are filled with fluid, cerebrospinal fluid, which Galen observed, to which very special properties were attributed. Remember, this is sort of post-Aristotelian views of how the body works. Galen discovered this fluid inside the head, cerebrospinal fluid, transparent, clear, perfect. So it was thought to be responsible for mental function and became known as animal spirit from animal, the Latin animal. So although Buddhist certainly talks about animal spirits, so I shall see in a second, he also attributed importance to the tissue as well, which of course, you know, now actually does the job. So just a few quotes from this remarkable book. The cerebrum is the primary seat of the rational soul. Animal spirits are the fluids, which he thought percolated through the tissue, right? For the various acts of imagination and memory ought to be moved within certain distinct, limited and bounded places, which I think must mean that he thought that different bits of the cerebrum were doing different things. Direct evidence for that, that's certainly a dominant idea. And then this phrase, which with a little stretch of faith, could mean something very interesting. These folds, he's talking about folded surfaces of the brain, these folds or rollings about are more and greater than man than any other creature, right? To which, for the various and manifold actions of superior faculties. So again, I think he's saying that these various superior faculties require different folding structures. But they are garnished with an uncertain, as it were, fortuitous series. He'd seen that in different people's brains, the folds were slightly different from each other. That the exercises of the animal function, the mind function, might be free and changeable. If an interest in the possibility that the brain is capable of changing and reorganizing itself, it's too much of a leap of belief to think that Willis was suggesting that. But the reason why brains differ between individuals is because they're free and changing in their structure, depending on how the different mental functions are being used. And that's going to be an argument that I'm trying to pursue. But if we leap to the 19th century, by the middle of the 19th century, or toward the end of the 19th century, it's a nice 19th century picture of the brain, neurologists would have recognized a number of distinct specialized areas, particularly for controlling movement of the body and analyzing the senses. So, from Hughes-Jackson's observations and others, that there's a strip in the middle of the brain, 3 central gyri goes down to control the motor neurons which drive the muscles. So damage in that region, for instance, produces weakness and uncoordinated muscle movement. Directly behind it, there's a sensory strip, parallel and posterior gyrus, which receives sensory information from all the body, and from the muscles, tissues, joints, and everything. And these two are interconnected so that incoming sensory information can generate movements. And the back, very well-known visual area. Virtually all of the fibres that come in from the eyes that are heading for the cerebrum end up innervating that region, which will look right at the very back here. So again, damage can cause blindness, and I'll talk about that in a moment. And then it will strip the top of the temple, though, responsible for hearing. That's how it is. You can see that an awful lot of the human brain, at least, isn't doing those things. And in fact, I suspect that since about 10% had clear functions attributed to it, this is probably the origin of that neuro myth that we only use about 10% of our brains, and somehow we could turn on the rest of this stuff and might be able to be much brighter. In fact, it was called silent cortex, as well as sometimes called association cortex, the rest of it, for a long time. The vague notion that the real things were being, the hard things were being done here, and maybe the rest of it really tried to associate them somehow and elevate cognitive function. Well, one of the ways in which cognitive function can certainly be elevated in humans is through language. And there's very good evidence that in those silent areas, there are regions that play a part in language. Broca's area, very well known descriptions in the late 1850s by Pierre-Paul Broca of individuals who had damage in that region of the frontal cortex, being unable to articulate. They could understand language, they could read, they just couldn't get things out, sometimes not a single word, and certainly totally ungrammatical. And then further back, in the 1860s, Carl Benneker described the symptom, another sort of aphasia associated with damage back there, which was almost the opposite, that these people with this condition could generate speech. In fact, you couldn't stop them generating speech. They just sort of had logorrhea, words coming out, and beautifully grammatical form, but totally meaningless. And they couldn't read and couldn't understand what was said to them, so at least in the extremes. So this leads to the notion that this area is responsible for interpreting language, and it's nicely placed for that. You see it not too far from the visual areas and from the auditory areas receiving information relevant to language interpretation, and it's very strongly connected with this region of Broca's area, which delivers speech. And by the way, that's very close to the sort of motor areas responsible for controlling the tongue, larynx, and all this sort of stuff. So, nice topology. But still, there's an awful lot left that doesn't seem to be doing things. This is mainly on the left side of the brain, particularly in most people. Okay, so we have localization of function as a principle, and the evidence for ever more precise, detailed, complete localization of functions has come from a variety of sources. Initially from painstaking work on animals, especially on monkeys, mapping out different areas of the brain with microelectrodes, showing that neurons in different regions respond to different sorts of things and seem to be involved in analysing different aspects of sensation and so on. But then very fully confirmed and to some extent extended by these brain scanning methods in humans. So I'll just show you some snapshots of the sort of things that can be done. This is using fMRI, functional magnetic resonance imaging, which is the most commonly used tool to look at human brain function these days. Basically what you do is you put it in a scanner, and the method measures local blood flow. It's really what it's measuring, water distribution, local blood flow. Tiny changes. The signals are microscopic, so a lot of this is all very clever analysis of tiny signals. In response to different things that happen to the person in the scanner. They're lying in the scanner, they have this terrible hammering noise, they might be uncomfortable or whatever. Those things are happening to them. But if you add something else, like looking at a